So chromosomes, our DNA, can be damaged in a couple of different ways. So remember that your chromosome, that sausage-shaped structure, right? A chromatid, double sausage chromatid X-shaped thing. It's just a long, continuous string of coiled up, packaged up DNA, right? And that DNA, again, is a long, continuous string, okay, and can be damaged, okay? So we talked about the types. We said there's direct and indirect types of hits. Depending on where the hit happens, where the damage happens, the DNA could be completely fine, okay, as in completely repairable. There can be some damage, or there can be large changes to the DNA's code depending on where these hits happen. So two broad types of damage. Main chain scission, as in scissors, as in cutting, okay, are breaks in the sugar phosphate rails. So those are breaks along the length of the DNA, okay? Do the rails carry any genetic information? Like, is there any inherent information in the rails? No. What do the rails do then? They hold everything together, right? They hold the information in one spot, right? So it's bad if you break both rails, okay? Because that like breaking a ladder in half, that breaks the ladder in half, right? It's less bad, less of a severe outcome if you damage one of the rails, but main chain scissions are damage to the rails. Rung damage is damage to the actual code, okay? Breaks in the nitrogenous base pairs that make the rungs. Now what we're going to worry about most here is something called a Frameshift mutation. So a mutation, in general, is just when the genetic code changes, okay? Mutations, so here, keep this in mind, right? Mutations are already happening, okay? They happen all the time, all right? Did I have a chance last time to play the video for you guys, the little 10-minute TED Talk video? I did, right? Okay. You saw how, like, the scale of that, right? That was what that video did a really good job is of showing you scale, right? It zooms in and shows you here's a strand of DNA, here's the copying mechanism, and then it starts to zoom out. And oh my gosh, look at how much DNA there was in just a single cell, right? So these frameshift mutations, these random breaks in the DNA and then reconnecting somewhere else, these are happening already, okay? Your cells do something called error correction, where 9 out of 10 of these errors, these frameshift mutations that happen, are correctable, okay? But that means there's 10% of them that are not correctable, and those are the ones that we worry about. All that we're doing with increased exposure to radiation is increasing the rate that these mutations happen. Again, they're already happening. We're increasing the rate at which they happen. Frameshift mutations are the problem because that changes the genetic code. Okay, so let's read from the left here. In the ladder analogy, to change the sequence of a genetic code, a frameshift mutation, both side rails must be broken, or an entire rung must be damaged. Okay, or at least one side of the rung, but there's some this is semi-repairable, but we'll say that that's a problem too. Both side rails must be broken or a rung damaged, okay? A single strand break, like in A on the left here, is said to be repaired. Would you like that to be... Can you see me now? You probably want to see the screen. Okay, so a break in just the single side rail is repairable, right? The body can come along and put in more of these sugar phosphate molecules to repair that. There is no break in the chain there. But double rail damage or rung damage can change the code. We call those frameshift mutations. So damages to the rails. Two types of damage to the rails: single strand or double strand breaks. Single strand breaks are repairable. Occasionally, the repair can be misrepaired and change the code at a single point, but more than likely these single strand mutations are repairable. The double strand breaks. These are what we're going to call hits. Okay, irreparable according to most people who talk about these things, most authors, and these can cause a frame shift. Double strand breaks are more likely to occur from direct effects than indirect effects. So hearing that, double strand breaks are more likely to occur from direct effects than from indirect effects. An indirect effect would be less likely because it would require two molecules, one to hit each side of the frame, where a direct effect can come from a single photon or a single particle hitting both rails. So if a double strand break is more likely from a direct hit than an indirect hit, is a double strand more or less likely than a single strand break? Less likely, right? Because direct hits are less likely, right? So good. So you can form these comparisons, right? These ways to talk about one versus the other. Rung damage, rung breakage, separation of the two base molecules can be repaired, okay, because they're held in place. Because if you break one side, the other side is still there, and you learned earlier, right? Let's jump back here. Okay, so I don't know that everyone necessarily remembers it. Find my location here. Thiamine can only bond with adenine. Guanine can only bond with cytosine. So if you break off a cytosine, okay, that's a rung that gets damaged, a part of a rung, then only cytosine can go back in that spot. Okay, so this is potentially a repairable thing. Some rung damage is potentially repairable. Okay, however, a loss or change of the base, okay, if an entire rung is changed, that is irreparable. That creates a mutation, a frameshift mutation, and that's going to change the code. That's going to build a protein that is not the right protein. That's going to cause the cell to build either not enough of the protein, or build the protein plus some extra, or not build the protein at all. Okay, that's a frameshift mutation. So here's a picture. Okay, rung breakage, which can be potentially repaired, or a loss or complete loss or change of a base pair, that is irreparable, the complete loss or change of the base pair. A mutation, just as a reminder, is any unrepaired changes to the genetic code. If the changes are repaired, then there's no mutation. Okay? So some reminders that I have said already, but let's keep it on the table. No mutations are unique to radiation. What does that mean?
That these mutations can happen whether you use radiation on whatever's mutated or not. It could just happen naturally.
Yeah, these are occurring all the time, right? We started off, one of the first things we discussed was that DNA is not a perfectly stable molecule, right? This is why and how evolution can take place. If DNA was a perfectly stable, with perfect fidelity molecule, it would just copy itself, and every copy would be an exact copy. Okay, that's not possible with DNA because it's just too large, right? The molecule is just too big to copy with perfect fidelity, so it changes randomly all the time, right? This is why cancer exists, right? This is why genetic defects, there's already a background percentage of genetic defects, okay? Mutations are there, they are random, they already happen. Radiation is not unique in that way. Radiation is unique because we can control it, okay? We cannot control the already occurring background rate of mutations, right? We can do things like improve people's quality of life, improve their diets, and those are going to help, but mutations happen anyways, okay? Radiation increases those. Most mutations are recessive, as in they're not going to be expressed, right? As you hopefully got in context that the whole DNA strand is not used to build your genes, right? Your genetic code. Those genes that are expressed that make you, they're not all expressed all the time, okay? Some genes, some traits are recessive traits, and those recessive traits are parts of the genetic code that are not going to be accessed by the cell. And if those are damaged, if those mutate, then there's not a big problem until down the line in one of your offspring, that mutated gene expresses itself, right? That mutated gene is activated, and if it's a cancerous gene, we call them oncogenes, and those oncogenes, can be recessive, and then eventually one of your offspring expresses that gene, and that offspring that has that genetic defect, that genetic problem. And the graph that you would chart, the graph that you would plot with this is a linear, no threshold graph, saying that any amount of radiation, no matter how small, may cause a gene mutation, and the incidences within a population would increase proportionally to the exposure the whole population receives. You give a population more medical x-rays in a year, you're gonna have more gene mutations. Okay. How's everyone feel about that? Anyone have any questions? I'd be happy to spend more time here. Okay, all right. Because now we're going to move on. So our next section here